Okay, in this screencast, we're going to be looking at the Calvin cycle, also known as the light-independent reactions. So the light-independent reactions, the inputs is carbon dioxide, and the outputs are going to be sugars. And we're going to take that carbon dioxide and basically build a bunch of organic molecules out of it. And we use the energy that came over from the light reactions, we use the ATP and NADPH to piece together those carbon molecules to make the sugar. Okay, now in this reaction, there's three phases. There's the carbon fixation stage where we're going to actually bring the carbon dioxide and fix it into a molecule that's part of this cycle. Then there's going to be a reduction phase where a bunch of energy is used to reduce the carbon molecules, or the sugar molecules within the cycle. And we're going to actually produce a triose phosphate, which is going to be our final product. So we get a triose phosphate. It's a 3 carbon sugar. And then finally, we're going to regenerate the initial carbon dioxide acceptor, which is called Ruby P, or ribulose biphosphate. So there's 3 steps: carbon fixation, reduction, and then regeneration. All right, let's look at those steps in a bit more detail. All right, so now in this process, we are going to we're going to make a triose phosphate. We need 3 carbons, so that means it's going to take 3 carbon dioxides to build that. So you can see here's a 3 here. We're going to have 3 carbon dioxides. So imagine this cycle happening kind of with 3 wheels happening at the same time. Okay, so it's going to take kind of 3 wheels turning together of this cycle to make one 3 carbon sugar. So the carbon, those 3 carbons are going to be attached with an enzyme called Rubisco. Okay, so the carbon dioxide is going to take, or the Rubisco is going to take carbon dioxide molecules and attach it to a Ruby P molecule. Okay, and so this Ruby P is a 5 carbon sugar, and the carbon dioxide obviously is a 1 carbon sugar. So we start with 3 carbon dioxides and 3 Ruby Ps. As I said, 3 are happening at the same time. And that is going to attach it to make a 3 short-lived 6 carbon molecules. Okay. Those 6 carbon molecules will split pretty quickly. So we go from 3 6 carbons to 6 3 carbon molecules. Okay. So that's the carbon fixation stage. Then we're going to go into the next phase, which is the reduction phase. So we know reduction is gaining electrons. We're going to go from these 3, I should actually tell you the name of those sugars. These 3 carbon sugars, very important, are called GP, or glycerate 3-phosphate, okay? Now, these GPs are going to be reduced in the next phase. All right, so now we're going to go from these 6 GPs, and they're going to be reduced. And they get reduced down here with the NADPH. So we've got 6 ATPs are going to come in, and then 6 NADPH will come in and reduce it. So it's going to be phosphorylized and reduced. And finally, the final product is we're going to actually get 6 of these molecules called triose phosphate. I realize on this it calls it a G3P, which when you go to university, they'll probably call it that, but IB likes to call it triose phosphate. So we go from the GP and we get 6 ATPs, phosphorylizing it. We're going to get 6 NADPHs, reducing it, and the product is going to be 6 triose phosphates. Okay. Of those 6 triose phosphates, one of them is good to go. That triose phosphate can be used to make glucose or other organic compounds. The other 5 are going to be reconfigured next back into the Ruby P. Okay, so we put in 3 carbons here and we got 3 carbons out here. So 3 carbon dioxides, we get 3 triose phosphates. All right, and then the last phase, these triose phosphates are going to be, you have 5 of them, so that's 15 carbons, are going to be regenerated into 3 5 carbons. So we went from 5 3 carbons, right? 5 3 carbons into 3 5-carbon molecules of RuBP. And look, it takes 3 more ATP to do that. So that regeneration of the CO2 acceptor, RuBP, takes a little bit more ATP. And that's why when I had mentioned when we did the light reactions that sometimes you get that cyclical photophosphorylation, and that you just need a little bit more ATP anyway, so that kind of works out. All right, 3 carbon dioxides enter. They're fixed by Rubisco to RuBP, right? And then that breaks and becomes 6 GPs. Those 6 GPs get phosphorylated by 6 ATPs. They're reduced by 6 NADPHs, and the final product is triose phosphate, 6 of them. 1 of them will be used to make glucose and other organic compounds, and the other 5 will be regenerated by 3 more ATPs back into 3 RuBPs, so the cycle keeps going.